After the thermocycler program is done, we need to see if PCR amplification of our unknown sample was successful before sending it for sequencing. To do this, we're going to use a visualization technique called gel electrophoresis. First, we add loading dye to each of the three reactions. This makes them denser and helps them sink for gel electrophoresis, and also adds a colored dye that allows us to watch as the samples progress through the electrophoresis gel. In this case, we're adding 10 microliters of 6x loading dye to each of our 50 microliter reactions. After loading dye has been added to all three reactions, it's time to load the gel. But first, we'll add a small amount of something called a DNA ladder. A DNA ladder contains many DNA fragments at multiple different known sizes, and helps us measure and estimate the sizes of DNA fragments in our PCR reactions. The DNA ladder also has a dense loading dye, so like the PCR reactions that we'll add next, it sinks into the wells made in the agarose gel, and is ready for gel electrophoresis. Next, we'll add the three reactions from PCR. First, the reaction with our unknown sample. Second, the reaction with our positive control, the known soft rot bacterium. And third, our negative control, which didn't have any DNA template added to it. The lid is closed on the gel electrophoresis box, and a power supply connected to the box is turned on. This creates an electric current that runs through the gel box, from a negative electrode at the top of the box to a positive electrode on the bottom. You can tell the current is active by the number of bubbles produced at the negative electrode. The electrical current causes negatively charged DNA molecules and the loading dyes to move through the agarose gel. Because the samples are initially loaded into depressed wells in the agarose gel, they are pulled horizontally through the agarose gel as they move towards the positive electrode. Agarose, much like gelatin, is made of a number of microscopic filaments that give it its rigidity. Smaller DNA molecules are able to more easily pass through the agarose filaments than larger DNA molecules, meaning that they travel further over the same unit of time, and causing a size separation on the agarose gel. This can be visualized using DNA stains that react to UV light and UV photography. In this example, the DNA ladder has a number of fragments in increments of 100 DNA base pairs, with the smallest fragment being 100 at the bottom. We can see that our unknown sample produced a band at about 300 base pairs, which is the same size as the band produced by the positive control. We can also see that there are no bands in our negative control, meaning no DNA was amplified. This suggests that the DNA bands really did come from our samples and not from some kind of contamination in our PCR reagents. Now that we know our PCR worked, we can order sequencing at the Cornell Institute of Biotechnology core facility. The core facility offers a number of molecular services, but we're going to select single-tube DNA Sanger sequencing. Here, following the facility's instructions, we'll enter the details for each of our samples as appropriate, including the names for each sample, the source of the DNA, and the approximate length of the DNA fragments as estimated from our electrophoresis gel. Once everything's in order, we can submit the order, then process and submit our PCR reactions per the facility's instructions. Sequencing results arrive in about 2 days.